Hi, my name is Taylor Upton, and my topic is biotechnology in dairy cattle production. According to the California Department of Food and Agriculture, dairy products such as milk are California's #1 commodity. Dairy products provide us with many nutrients such as calcium, potassium, vitamin D, and protein. Overall, agriculture is extremely important in our everyday lives. Improvements in biotechnology and genetics of dairy cattle are very valuable and important in the dairy industry. I have always been very interested in cattle. I work on a farm where I work with cattle of all sizes every day. I am an animal science major, so this topic is very relevant to my field of study. I find this topic very fascinating and I enjoy researching it. Today I will be discussing my 3 themes, which are reproduction, milk, and animal welfare. My first theme is reproduction. How has biotechnology improved the genetics of dairy cattle's reproductivity? There are many ways in which dairy cattle can be genetically edited to produce offspring with desirable and superior genetics. These desirable traits include milk production, growth, feed efficiency, disease resistance, body structure, muscle, and fertility. Chris Proudfoot, Daniel Carson, Rachel Huddart, and Associates published an article in 2014 including a study that they conducted. In this study, they utilized ovum pickup, in vitro fertilization, and zygote microinjection to produce gene-edited calves. This allowed them to insert desirable alleles into a target breed or genetic line. This study produced 6 calves, 4 alive and 2 dead due to calving difficulties associated with twinning. Of the 4 live calves, there were 3 bulls and 1 heifer. The 3 bull calves expressed the modified genes, however, there was no observable difference in the heifer. This is a picture from the article. It shows the live-born bull on the left, the heifer on the right, the phenotypic differences between the bull, which is on the right, and the heifer, which is on the left. You could see major differences. In the journal article Major Advances Associated with Reproduction in Dairy Cattle, the authors Moore and Thatcher describe many other forms of improving dairy cattle genetics reproductively. These mechanisms include superovulation and embryo transfer, in vitro production of embryos, sex semen, cloning, and transgenesis. Superovulation and embryo transfer allows producers to obtain multiple offspring from genetically superior females by transferring their embryos. In vitro production of embryos allows producers to generate large numbers of embryos with improved genetic potential from oocytes isolated from the ovaries. Sex semen is used in altering the sex ratio in favor of heifers, which is a great advantage for the dairy industry. Cloning is used to reproduce animals by nuclear transfer for the production of an unlimited number of genetically identical offspring. Transgenesis allows many beneficial traits to be added from other species. This is a diagram of sex semen, sperm labeled with a nucleic acid stain and sorted on a fluorescence activated cell sorter. X-bearing sperm contains more DNA and fluorescence and glows more, which allows them to be separated from the Y. Then back to this one is a diagram of cloning. So the donor cells, the transfer donor cell in the needle is inoculated into the oocyte, cell fusion and activation, the embryo, and the new cloned calf. My second theme is milk. How can biotechnology be utilized in milk? Biotechnology is not only in food processing and enhancing dairy product properties such as taste, shelf life, and nutritional value. Dairy products can be genetically modified to further improve and benefit human consumption and human health. In a study conducted by Jabed Wagner and Associates, they validated targeted miRNA expression as an effective strategy to alter milk composition and other livestock traits. Milk from dairy cows contains beta-lactoglobulin, or BLG. This is a major allergen, so the purpose of this study was to decrease its levels in milk. Hydrolysis was previously used, but it left residual antigenicity. It caused a bitter taste, and the processing is costly. This study produced a transgenic calf whose milk contained no detectable BLG and more than twice the amount of casein milk proteins. This study provides the opportunity to fully investigate the impact of the associated milk composition changes on nutritional and processing properties and will provide new insights into the still obscure function of BLG. The objective of the study conducted by Bernal, Santos, O'Donnell, and Associates was to increase the N3 fatty acid content of milk fat using stearidonic acid or SDA enhanced soybean oil or SBO derived from genetically modified soybeans. N3 fatty acids such as SDA are very beneficial to human health. They are essential to growth and development. They are important in the prevention of chronic diseases including cardiovascular disease, inflammatory diseases, and neurological disorders. 3 Holstein cows were used in this study. One was the control, which received no oil infusion. The second one received infusion into the abdomacum of SDA-enhanced SBO, and the third received a ruminal infusion of SDA-enhanced SBO. The results demonstrated that the abdomacum infusion of SDA-enhanced SBO resulted in a 5-fold increase in the N3 fatty acid content of milk fat. Unfortunately, the results were affected because one cow suffered a physical injury and had to be removed from the study. My last theme is animal welfare. How is biotechnology utilized to improve animal welfare? Animal welfare can be improved through gene editing in many ways. This includes genetic disease resistance, genetic polling, as well as genetically modified dairy cattle feeds. In the article Engineering Disease-Resistant Cattle, the author discusses a study conducted to prevent mastitis in dairy cattle. Mastitis is a disease of the mammary gland that costs the United States dairy industry approximately $2 billion every year. This article discusses the utilization of transgenic dairy cows that express lysostaphin in their mammary epithelium. Lysostaphin attacks the mastitis pathogen. These transgenic cattle resist the mastitis pathogen. Their milk, which contains antimicrobial peptides, kills the bacteria. This study was conducted in hopes of expanding the ability of creating transgenic cattle and improving disease resistance. McConaughey, Hotzel, and Associates discuss genetically polling of dairy cattle in their article. Dairy cattle typically grow horns, and these horns are often removed at a young age using a procedure called disbudding. Disbudding or dehorning is done to reduce the risk of horn-related injuries to farmers as well as other animals. Gene editing can be utilized to spread this polled gene. The genetic modification is utilized to improve animal welfare and overall benefit the animal rather than the consumer. These genetically hornless animals do not need to experience the painful procedures used to remove the horns or horn buds. This is a picture of the little horn buds. They say that if you remove them when they're younger, they're small horn buds and easier to remove. And then when it's actually called dehorning is when you wait till they're older and they have full-size horns. My topic is how is biotechnology utilized to improve and increase production in dairy cattle. I discuss the themes of reproduction, milk, and animal welfare. Some major drawbacks in my research include negative public attitude toward genetically modified dairy cattle, expensive processes, and mortality. Overall, biotechnology has greatly benefited the dairy industry. However, there is still so much room for vast improvements. The articles that I've discussed today have greatly contributed to the advancements of biotechnology in the dairy industry. I believe that these genetic modifications are extremely beneficial and important to agriculture in our everyday lives. These tools will accelerate the utilization of engineered livestock for biomedical and agricultural applications. There is so much opportunity for biotechnology to expand in the future. Here are the links to the articles that I used. And thank you, and I hope you enjoyed my presentation.